Hi, everybody. We're going to get started with the second lecture for week 2. We just covered the cell structures and their functions, and now we're going to get into a little bit of molecular biology. So I'll go ahead and share my screen and bring us to the lecture. I will be moving to the whiteboard for part of this as well. So the learning objectives for this particular mini lecture, the first is that I'd like you to be able to explain the DNA replication process. We're going to do an overview of it, so I'm not going to get into great detail about all of the steps that take place in DNA replication, but hopefully give you a good kind of overview of that process. In addition, we're going to talk about the 2 steps of protein synthesis, how cells make proteins, but also I want you to keep in mind how those structures within the cell could be utilized by the virus to make those proteins as well. I will explain where and when you would expect to see a cell go through DNA replication and protein synthesis, where those processes occur, and when the cell would do that. Lastly, I'd like to explain why viruses need the machinery of their host in order to replicate their own DNA and also make their own proteins. There is vocabulary. We have replication, enzymes, nucleotides, semi-conservative, gene expression, which can also be referred to as protein synthesis, amino acid, and peptide and protein. A peptide is just a small version of a protein. So we're going to start out talking about DNA, and DNA is an abbreviation for deoxyribonucleic acid. It's a molecule that when we have nucleotides that are joined together, we form these long double helixes of DNA. DNA's whole function is a code with instructions for all living organisms. So every cell in your body contains DNA that has the code for those cells to do the processes and structures that they need to be that cell. As I said, every cell contains all the instructions that we term the genome. These DNA molecules are made up of nucleic acids or nucleotides, and each of these nucleotides has what we call a base. Together, the sequence of these bases, so it's like a computer code that the cell can read, and they can read these base letters as part of this code that gives them their instructions. So I want you to think about DNA as a double-stranded helix of nucleotides. I'll go to my whiteboard and draw just kind of a basic version of this. Each of the base on one strand will actually bind with the second strand through what is a complementary process. So if we have an adenine, which we would have an A is representative of an adenine, it would bind always to a thymine. And if we have cytosine or a C, it will always bind to a guanine or a G. And so this also allows DNA, when it is separated, the 2 double strands, to be a template in which we can do DNA replication, or that we could also do protein synthesis. So DNA replication is the process where that DNA, that double helix, can be copied. And this would happen during cell division, so that if one cell is dividing into 2 cells, each of those new cells then would receive a copy of the genome. I'm going to bring us over then to the whiteboard, where I'd like to draw a little bit of this out and kind of have you thinking about it. Please note there will be videos also, and if you have questions, this is one that if you haven't had biology in a long time or you've never had it, you may want to stop by my office hours if this is confusing. So we're really talking in this case about DNA. And I'm going to do kind of a picture of what DNA would look like. Try not to be too complicated, but give you a good idea. So DNA is made up of a ribose, which is a sugar. So I'm going to put this is ribose, and it's a sugar. And it also has connected to it a phosphate. And then off this side, we have a base. And this would be either the adenine, thymine, cytosine, or guanine. And what ends up happening in DNA is this is just, so this whole structure here is what we would call a nucleotide. And it is made up of a phosphate plus a base plus a sugar. And that would be the ribose. So the really cool thing about this is the nucleotides, as we begin to link them or bond them together, what we end up having is you would have many of these structures, right? And I'm going to do this with maybe a letter attached, phosphate, and then we would have another one that would be added on, and maybe in this case, it's a cytosine that's added. You'd have a phosphate, and these will begin to form kind of this chain, if you will. And this is basically what we would expect our DNA to look like. But I said it was a double-stranded helix. So what you want to think about is on the other side, there would be, oops, excuse me, I'm going to erase that line. It's always hard to do this other strand because it's kind of upside down, if you will. Basically, the same form. We're going to continue over here having the exact same thing, but hopefully you've noticed it's in the opposite direction. So if we were to look at our sugars, our ribose, what you might see is that the sugar on this side, they're all pointing in this direction. And in our second strand, they're all pointing in this direction. And if you've ever heard it referred to, this is what we refer to as anti-parallel. And what that means is that these two strands run in opposite directions, but they do connect in the middle. And so I'm going to put a base here. We said A is complementary to T and C is complementary to G. So if we have an A here, that means on our opposite strand, we're going to have a T. And down here, when we have the cytosine, we're going to have a guanine. And then over here, we have a guanine. That means on the other side, it would have a C when I add that other sugar. And this, if you're starting to look at this, what I hope that you can see about this is that these letters can form a code. Basically, these letters can be read, and these are what would be the code or instructions that the cell can read or the enzymes of the cell can read. The other thing that I'd like you to note about this is that if we have one strand over here and we want to make a copy of this double-stranded, if we basically unzip these two strands from each other, we have templates where we know what the opposite strand should look like, right? So if we were to look over here and see a T, we'd know that on the opposite strand would be an A. Or if we looked here and we saw a G, we'd know it was a C. So all we really need is one half of this molecule in order to be able to tell what the other half would look like. And really what you want to think about is when this is kind of, if you were to zoom out, what we would see is basically this kind of looks like a ladder. And if you've ever seen DNA, you kind of know what I mean. And what we would think about this is that the rungs of the ladder, the part that is crossing over in between is the bases that are paired together. And these rails of the ladder, the sides of the ladder, are the sugar and phosphate. And usually this looks twisted. So if you've seen it as a twisted molecule, that's what we're thinking about. But not only is the DNA anti-parallel, right, running in the opposite direction, but it's also what we call complementary. And if you've heard that or you've seen that when you've gone through my notes, the complementary is just referring, this is referring to the fact that these are complementary, these base pairings. So hopefully that gives you kind of a quick idea of what DNA looks like. We'll go back and talk about when it's replicated and when we use it for protein synthesis. But I do want to just briefly talk about that. So DNA actually plays a role as a template. And there's 2 times that it does this. And so I kind of want you to think about DNA being a template with all that information. We can kind of divide that into 2 halves. One of them is DNA replication. So when the cell maybe is going to go through cell division, it needs to make a copy of its DNA. It will do so doing DNA replication. So this is the copying of DNA, okay? The other thing, though, that DNA is used as a template for is what we call protein synthesis, and I'll get to that in just a minute. But protein synthesis, DNA has the information in which a cell can make proteins. And so ultimately what we get out of this is proteins are produced. So let's go back to the slides and kind of go through this on my slides. So hopefully some of this is going to now sound a little bit like what I covered on the whiteboard. We have DNA as a double-stranded helix of nucleotides. Each base on the one strand will bind with a complementary base on the other strand. And it is due to this complementary characteristic that each strand then can be a template during DNA replication. And we want to think about DNA replication as that process where the DNA is copied during cell division. Remember? And so when one cell divides, we have to make a copy so that each of those two cells will have a copy of the instructions. So this is kind of giving you another view of what this looks like. So on the left-hand side here, we have that double-stranded DNA. Think of it as that ladder that I drew. So on this side, remember how the sugars were all pointing upwards. You can picture them in the rail over here. And on the complementary strand, the sugars were all pointing down. And so that would make them anti-parallel. And then in between, you can see the rungs of the ladder. We have that pairing that I talked about. Adenine to thymine always, and guanine to cytosine. You're always going to know if this half was covered or gone, we would still know what should be the complementary base. And so what we see when we have DNA replication is that if this is the parent cell and it has that double-stranded DNA, because each side of the strand, the double strand, can be a template, the two new strands are what we call semi-conservative. One half of it will be the old or parent. So if you were to look over here, we can think about it as this strand on the left. And then it will have all new nucleotides that have come in that are complementary to make up the other half that's missing. And we have the same thing over here with this maybe being the parent strand and then new strand that's complementary. And that's basically what goes on during DNA replication. And I want you to think about the idea that this is taking place in the cell before cell division. But if a virus is infecting the cell and it needs its DNA replicated, it will utilize all of the same functions that happen in the cell. So it will be able to utilize the enzymes and functional proteins that direct this process. So viruses basically steal those enzymes in order to replicate their own DNA when they infect a cell. The next part of this is talking about protein synthesis. And if you haven't had chemistry or biology in a long time, I want you to think about the fact that proteins are what we really think of are the workhorses in the cell. They generally are what get reactions to go forward quickly so they can instigate reactions or get reactions to occur. They play tons of different roles of transporting things, of making reactions move much more quickly. And so cells need to have proteins being produced all the time. So protein synthesis is something that is occurring all the time in a cell. But I want you to stop and think for a minute about where we talked about proteins when we talked about structures of viruses. So my hope is that while you thought about that, you remembered that proteins are used to make the capsule. So those capsomeres are made up of proteins, and that's what makes up the capsid around in a virus, but also the spike proteins that we'll talk about in the next chapter. Many viruses have what are called spike proteins. Again, viruses can't make these on their own. So in order to make these proteins that they need, they depend on the cell to do this protein synthesis. So I want you to keep in mind that protein synthesis has two steps, the first of which is called transcription. And basically part of the DNA code will be transcribed or copied into a mRNA. mRNA is similar to DNA, but is single-stranded. And that mRNA or messenger RNA will take that information from the DNA to a ribosome where it will be translated or made into a protein. So we're always thinking about in protein synthesis that it starts at the DNA. At the DNA, a small piece of mRNA with a message will be made, and that then goes to the ribosome where the protein can be made. This process requires the gene, the part of the DNA that's going to be expressed. It will also require an enzyme called RNA polymerase, and it will require mRNA or messenger RNA to carry that message from the DNA to the ribosome. Obviously, ribosomes then will be needed. And the tRNA is actually what will be bringing in the building blocks to make the protein, which are called amino acids. So with that, I am going to go back to the whiteboard and kind of walk us through this again in kind of those broad terms. So we talked about DNA replication, copying of DNA. I do want you to keep in mind, this occurs just prior or just before cell division. But in a cell, protein synthesis occurs all the time because cells need proteins, okay? And when we think of protein synthesis, this will often be called gene expression as well, so don't be surprised if I use those interchangeably. And the reason why that is I'll show you what this looks like when we think about it on kind of this another bird's eye view of this. Now, I only have room on my page to draw kind of a short piece of DNA, but we're going to talk about this. So if this is the DNA, remember the rungs of the ladder or those bases? If this is the DNA, I want you to picture, though, at home, if you will, that that ladder goes up through the top of your computer screen and the bottom of it goes down to maybe the floor in your room. And we can think about that there are what are called genes. So we might have a segment of DNA, just a segment, and maybe there's another gene, right, that goes up here that's another gene. But these genes are sections of the DNA that carry the information for one protein. So I like to think of DNA kind of as a cookbook, that it holds all of the recipes of different meals that cell might need to make. And each recipe is actually a gene. And if you take those instructions in that cookbook, if you will, and you make the meal, that would be the protein. So when I think about that, with this, the gene, we can have an enzyme come in and it will open up the DNA and it will copy one side of that gene. So remember the A's, T's, C's, and G's? It will copy that and it will make a single strand that's complementary to that is called mRNA. And the mRNA stands for messenger RNA. And so this RNA basically has the information or the recipe from the gene to make one protein. And what's going to happen with that is that then will go to, I'm going to draw it like this, a ribosome. And when that mRNA goes to the ribosome, what ends up happening is it will be read by the ribosome, and the ribosome then will make a protein based on that recipe. Oops, there's protein. And I'm going to draw it sort of like this. A protein is a bunch of amino acids that are bonded together. Okay? So we'll think of that as made up of amino acids. Okay? And so this is really what happens in protein synthesis. Now, I said there were two steps to protein synthesis. And so I do want to say that we should put in what those two steps are. The first step is what we call transcription. And transcription is where we take DNA and we make mRNA basically from it. The second step then here, second step is called translation. And that's where our mRNA will be translated into a protein. Okay. And so this is what goes on in the cell all the time. It's always expressing these genes or making these recipes. But if a virus infects that cell, it can utilize this as well. It can utilize those enzymes that do the transcription portion, so it can make a transcript of its genome, of its particular genes, so that there's an mRNA that can be translated into a protein that the virus needs. And so that's a really important thing to think about, that the viruses utilize from it as well. So we'll go back and finish up the lecture. Let's move us to the next slide. So I do want to just get you to kind of think about the different molecular biology we just covered. So to kind of summarize this, we can have replication of DNA, and then we can have transcription and translation to make protein synthesis. And we want to think about this, that they have certain locations where these take place. So DNA replication and that RNA transcription are always going to occur in the nucleus, and that's because that's where the DNA is located in a cell. And you guys should have known that from earlier in this week of the things that we already covered. The translation, though, will take place at a different place than the nucleus. This will take place at ribosomes. And remember, ribosomes can either be found in the cytoplasm or they can be found attached to the rough endoplasmic reticulum that's also in the cytoplasm. So some quick summary: in eukaryotic cells, we have replication of DNA. That means we have DNA making copies into more DNA, and this occurs at cell division. A key player or key players in this are enzymes called DNA polymerases. We also have protein synthesis, and this would include the transcription and translation. In transcription, DNA is being transcribed into mRNA. Key player in this is RNA polymerase, an enzyme that can make mRNA from DNA. And translation, where we have mRNA that's being translated into a protein. Key players in this are the mRNA, tRNA, and ribosomes. Oops, sorry, jumped ahead there. So as we think about this, I do want you to go back and think about what the virus then would utilize in that host cell. And viral genome replication and protein synthesis require the machinery and enzymes that are found in cells. They don't have their own ribosomes or enzymes to do this. And so that's why they are obligate intracellular pathogens, is because they require that machinery and enzymes that are found in the cells. They need it to make new virions, because to make a new virion, they have to have a copy of their genome, and they have to have the protein synthesized for the capsid spike proteins, and then if they have any special enzymes that they themselves carry with them. That puts us on the checking in. I'm going to let you go ahead and pause this before I answer these questions. So let's walk through these. The first question is, what is the result of DNA replication? It's a copy of the DNA. Where does replication occur? That's going to be in the nucleus. What are the two steps of gene expression? Transcription and then translation. Where does gene expression occur? Transcription occurs in the nucleus. Translation occurs at the ribosome. Why do viruses require host cells to replicate? Viruses do not have the enzymes, nor do they have the ribosomes to copy their DNA or to make proteins. What cellular structures do viruses use to replicate? They use enzymes, ribosomes, That's pretty much it. All right. If you have any questions, please let me know. Thank you.